ahead and start.
Okay. Okay.
Yeah.
So, Pankaj, you please take care of letting people in, etc. I will now focus exclusively on the seminar. First of all, good evening, everybody, for joining us for this rather happy occasion. My student Tathagat Biswas will be defending his PhD thesis. Uh, so Tathagat joined before joining my laboratory. He did his BTech in electronics engineering from West Bengal Technical University, and then he actually was working for Accenture as an associate software engineer, and then he wanted to change career track and join a PhD program in biology. And obviously the first couple of years was a little rough for him, but then he picked up, and he has done his work understanding the role of three novel genes in the differentiation of vertebrate skeleton. Uh, he will be presenting his seminar. The Let me introduce the oral board to you. We have amongst us external examiner from National Institute of Reproductive Biology, Dr. Deepak Modi. And then we have from IIT Kanpur, from the Chemical Engineering Department, Professor Sri Shivakumar. And from BSB Department, Dr. Arun Shukla and Dr. Nitin Mohan, and myself, Amitav. So that's the oral board. The format at IIT Kanpur is that we will first have the seminar. Tathagat has prepared the presentation of approximately 50 minutes. And you should feel free to, because of this online format, it is difficult to ask questions in the middle. But even then, if hands are raised, I will try to recognize and see if the questions can be asked in the middle. Otherwise, at the end of the talk, it will be open for questions. And after question answer session is over, then it will be a closed door session, rather closed internet session, where we will request everyone else, including the candidate, to leave the room. Only the examiners will be here to decide on the defense, whether it is to the satisfaction of the committee or not. So with that, I would now request Tathagat to please start his presentation. Over to you, Tathagat.
Okay, so I'm audible, right? Yeah, so thank you for the introduction and welcome everyone to my defense. So today the title of my talk is A Study on Three Novel Regulators of Vertebrate Skeletal Differentiation. So when we talk about vertebrate skeletal differentiation, it's important that we note that there are two main mechanisms through which bone forms in a vertebrate system. So firstly, the craniofacial region, which is the bones around your skull, which are mostly flat and consist of immovable joints, they form through the process of intramembranous ossification, wherein the loose mesenchymal cells, they condense to directly form bone. However, the second mechanism that we have, which deals with the long bones. So these long bones are the bones of your appendicular skeleton, that is your hands, your legs, etc. So these form through the process of endochondral ossification, wherein the loose mesenchymal cells of the limb bud, they condense to form a cartilage model initially. And eventually this cartilage is replaced by the bone. So our lab has been trying to understand different facets of endochondral ossification, starting from very early embryonic development itself. And my thesis itself actually revolves around different aspects of endochondral ossification. So to make it easy, I'll give you a brief introduction into different steps and stages of endochondral ossification. So as I told you that initially there's a loose mass of mesenchymal limb bud cells. They condense together to form one continuous unit of cartilage. Here is a graphical representation. You can see in the limb bud, there is one single unit of cartilage. In this erstwhile single unit of cartilage, the first sign of discontinuity arises with the formation of interzone, which is a morphologically distinct band of flat cells. It is at this site of interzone that you have subsequent segmentation, and it's in this site of segmentation that you have joint formation, which is the movable joints, your hands, your legs for locomotion. So it should be noted that with segmentation, what was initially one unit is now divided into independent units, and these units, they become independent themselves, and then they grow, and these growing elements contribute to each and every element that you have in your limb, as you can see in the cartoon. I'll just digress here to point it out that interzone, which in turn specifies the location of the joint, also to some extent regulates the length of these bones. That is, for example, the femur would be the longest bone in our body, is kind of dictated by the location of interzone also. So coming back to the cartilage elements, so as they are growing, the cells inside the cartilage element, they also go through several stages of maturation. Initially, they're proliferative, and after stopping proliferation, they get into prehypertrophic state, and finally, they enter hypertrophy. It is at the hypertrophic stage that the cells grow in size. They secrete a lot of extracellular matrix protein, which are important for bone formation. There's also blood vessel innervation here. And it's at the central region that ossification starts, that bone formation starts. And then, as I told you, though initially from the center, finally the entire cartilage element is replaced by bone, leaving behind only the terminal ends, which are the articular cartilage, or the cartilage which is important for moving around. You have it in your knees, in your hands. So in my thesis, I'll be dealing with three different aspects of endochondral ossification. The first is induction of interzone, where I'll be talking about the transcription factor, BARX1, and its role. The second project would be about PRDX1, which interestingly is an antioxidant protein, and we will see how it's important for prehypertrophic state maintenance. And finally, I'll be coming to DPYSLC, which is a BMP downstream target identified quite many years back by our group. But now we will see how it's important for cytoskeletal regulation during skeleton development. So to start off, I'll be starting off with the earliest of developmental event, that is interzone induction. So what was known about interzone quite early is that it's essential for joint formation. Here, Holder, in a very elegant surgical experiment, he showed that when he removed a developing chicken wing limb bud's elbow interzone, he excised it out and stapled back the rest of the limb together. What he found, if he let it develop, was when finally the wing was formed, it developed without an elbow joint, thus making sure that we understand it's necessary for joint formation. But though we know that it's necessary, what we did not know for a long time was what is the origin of cartilage interzone. Initially, as you can see in 1940s, far long back, people thought that it's a fibrous tissue positioned in between two skeletal elements, and that also helped them explain the morphology of the tissue, which is flat. So it's squished in between two cartilaginous units and therefore flat. But with developing genetic tools, we realized that is not the case. So if you go with me through the slides, in the first panel, you see both of these are actually E13.5 mice embryos mean region. So in the first panel in blue, you have all the cells which are expressing collagen 2, which is a marker for cartilage. So femur and tibia are both here at this point in time are cartilaginous. However, if you look at the interzone, it's completely devoid of cartilage expression, that is called 2A1 expression. However, if in an adjacent section, which is like 5 microns away from this, in the second panel, you see all the cells which are blue are actually expressing LAG-Z. So I'm not getting into the details of the mice line, but what happens in this mice line is that all the cells which had a history of collagen 2 expression.
It's not audible. Your net connection, you please check it once. Suddenly the quality dropped.
Okay, just a second. Is it better now?
We can hear you very well.
Okay, so okay. It looks like for everything else it is fine. It is only my problem.
Okay, okay, fine. So as I was talking about that, this in all the cells which are expressing lags are actually cells which at one point of time expressed collagen 2, which is a cartilage marker. So this shows that interzone cells actually originate from a call to expressing population of cells. And therefore this cartoon, where from an initial contiguous unit, you have interzone cells, which are devoid of collagen 2 markers. Now, even we know that there are markers of interzone as well, like dedicated markers like GDF5, which are expressed exclusively in the interzone. What you see here is a digit interzone. So digits are fingers. So this is a chicken digit interzone, and you have exclusive interzone specific expression of the molecule GDF5, and so is autotaxin GDF5. These are markers of interzone. So if I put the points together, what we see is that the salient features of interzone are that they're flattened band of cells. They are actually emanating from and call to expressing population. And after being interzone, they stop expressing collagen 2. And finally, now we know they have their own set of markers like GDF5 and autotaxin. So what precisely happens is from an existing mass of contiguous cell, there emerges a new cell type, and that is the interzone, which loses its cartilaginous property. So the question becomes that what induces this induction of a new cell mass in the cartilage? So we wanted to explore that, though we weren't the first to think of this question. So before I get into that, it's important that you know that when we are looking for a molecule which might be inducing this new mass of cell, this interzone, it's logical that we go after molecules which are expressed exclusively in this region. So as I had already introduced, you know that GDF5 is one such molecule. It's expressed in the interzone. And if we assume that GDF5 is important for interzone formation, then ablating the function of GDF5 would lead to abrogation of interzone formation. And that is what scientists actually observed, that GDF5 mutant, if you come to this slide, if you look at my pointer, so GDF5 mutants actually develop the digits. This is a mice GDF5 mutant embryo without the interzone. It's one contiguous element, whereas the wild type in the same stage maps is having interzones and joints properly formed. So that was exciting that mutating or ablating GDF5 is not letting interzone form. But the reverse corollary of this would be that if we misexpress GDF5 in the cartilage, there would be more and more interzone formation. So that experiment was done by Storm et al. And what they found was actually the opposite. That when they placed a bead of GDF5, which you have here in this asterisk mark, it actually abrogated interzone formation. It stopped interzone formation. So before I get to the next slide, I'll just explain to you how to look at this data, because that will be helpful for me in explaining all the other data of mine. So in chicken, the problem is that each of the eggs or the embryos are independent from each other. And they are fertilized outside, and then they're brought into lab, and then they're incubated. So what happens is there is some stage variations amongst the embryos. So what in the field is preferred is that you have manipulation and non-manipulated region in the same embryo, or even better, if in the same tissue. So here they have used that idea. You can see that where the bead was placed was actually a developing interzone. So this region where the bead's effect can be seen, here the interzone is stopped. The interzone does not develop. However, the other region, which is out of the domain of GDF5's influence, and this serves as the unmanipulated region or internal control. So in here you find that there is proper development of interzone cells. So this concept of internal control, where one portion in the same tissue is affected and the other portion is not, would keep coming back in case of chicken manipulations again and again. I'll come to it again. But finally, about GDF5, what was observed was that it's not an inducer of interzone. A very similar molecule was Win14. So Win14 again was expressed in the interzone, much like GDF5. And when Kristen Hartman and Cliff Tabin, they decided to misexpress Win14 in the cartilage, they used a viral vector. Now, when you see this data, I'll again explain there is one caveat in using virus, and that is when you introduce virus in chicken, most of the times the infections are patchy. And hence you can see here, these are skeletal tissues in these two arrows here. One of the skeletal tissues are having a patch of infection. But this patchy infection also lets us have what I explained to you just a slide back, that a region which is unmanipulated and a region which is infected. So if you look at the skeletal tissue, which is having the patch of infection of Wnt14 virus, GDF5 and autotaxin, the markers of intozone, are overexpressed in this infected patch. Whereas the internally, what we call as internal control, this uninfected patch of skeletal tissue does not have such expression. Therefore, Wnt14 was capable of inducing the intozone markers. However, what we see is that there is no flattening of cells, which was the most conspicuous feature of intozone. And moreover, when Hartmann herself, she opened her lab and she knocked out Wnt14. By then, its name was Wnt9A. There was a change in nomenclature. We see that there is no such change or problem in segmentation per se. You have the wild type mice here, you have the knockout mice here, the segmentations are forming all right. Therefore, the question persists, what induces intozone formation? So we had a head start. Again, we were looking for a molecule which was expressed in the intozone, but fairly early. So Pratik, a previous graduate student in our lab, he was working on an mRNA screen of articular cartilage region. If you remember, I told you that articular cartilage is at the joint location. So while working on it, he found that BARX1, a transcription factor, is expressed in the intozone region. And a quick literature survey actually revealed that BARX1 is expressed pretty early. I'm not getting into details of these stages, but what you see by HH are hamburger, Hamilton, chicken stages. So they are all the developmental stages in chicken and how we label them. But long story short, what we realized is BARX1 is expressed in the intozone far earlier than what was believed of other intozone markers, and even before intozone is morphologically evident. The second line of evidence came from one of our collaborators' work, Terence Capellini. So he was working on enhancer elements of GDF5. So enhancers are DNA elements regulating the expression of a downstream gene, which in this case was GDF5. So if you look into one of those regulatory elements, say for in this case R2, which he named as, what he found was that it is studded with a number of BARX binding domains. There's a S1 to S8. And if he was driving the expression of LAG-Z under the reporter R2, as you can see here, again, if you follow my pointer, in the first panel, there is exclusive expression of LAG-Z, again in blue, in the intozone or joint region. Here you have R2. However, if you mutate the BARX binding sites, that is S7, S8, or S6, you see that there is complete loss of LAG-Z expression in the intozone domain. And the third line of evidence actually comes from our own lab again. So Upendra was working on joint location, and I won't be getting into details of it because he will be giving his open seminar soon. So what he could do in one line is that when he changed retinoic acid and FGF gradient, when he manipulated this gradient, he could change the location of the forming joint. And what he observed was that this location change in the joint actually was preceded by a change in location of BARX1 expression. In the control, you see BARX1 is here, and you can see how distal it has moved after it's manipulated RNA and FGF. So putting the three things together, that it's expressed, BARX1 is expressed much before intozone is morphologically or molecularly apparent, and that its binding domains are important for intozone marker expression, and finally that it precedes any shift in intozone or joint formation, we thought it would be exciting to look into BARX1. And to investigate the role of BARX1, I used a technique which was used also by Kristen Hartmann and Cliff Tabin. That is, I cloned full-length BARX1 into an avian viral vector. So this is a replication, competent avian sarcoma virus. And after I've cloned BARX1 into this vector, I electroporate the embryos at HH14. So this is day 2. It's just 2 days after fertilization. And then I harvest these embryos at HH34, which is day 8 of chicken embryonic development. And by then, most of the bones and ossification and morphological developments has happened. So here you see a representative image of stage 2 embryo. In the flash sign is just above a presumptive limb bud. If you take a cross section here, you will see that there is a lateral plate mesoderm, which has a lumen in between. And what you see in blue, that is where we inject our DNA. And then we put our electrodes and we electroporate. And if you look at it 8 hours from then, you see this beautiful GFP positive limb bud in the chicken embryo. Now, when I harvested these at HH34, what I found was that all the embryos had tremendous difference in their morphology altogether. They were malformed, they were short, and investigation, like if I looked into the light microscopy, I saw that there were a lot of segments in the proximal elements of these limbs, which are malformed. So I sectioned these tissue, and what I find is that indeed, so here you have Arches Barix1 limb, which has number of segments as opposed to a contralateral. Again, this serves as a stage matched control. So the contralateral limb is devoid of any such supernumerary segmentation, but what we find here in Barix1 infected one. So this is a safranin O staining. So safranin O stains for the cartilaginous matrix. It stains the proteoglycans of the cartilaginous matrix. So this is also a proxy for collagen 2 staining. I explained this because I'll be coming back to it again. So now when you look into the molecular signature, the first thing that I do is look into the extent of Barix1 expression. So in green, you have all the regions and cells which are infected with Barix1. Now, if you remember, if it is interzone, then collagen 2 expression must go down. And if I look into collagen expression in red here, I see indeed the region which was heavily infected is having downregulation of collagen 2. And not only the heavily infected patches, if I take you to one of these small patches and magnify that, you would see that all the cells which are having infection with Barix1 is actually downregulating collagen 2, and even there is conspicuous flattening here. The flattening is more clear if you look into a safranin O stain histological section, where you find that the region where they've lost safranin O, which I told you is also a proxy for cartilaginous region. So these regions are no more cartilaginous because they've lost collagen 2 expression, and they're far flatter than the, again, internal control region, which are called to positive and bulbous. Now, what about the markers? So again, in the infected region, which is downregulating collagen 2, leading to flattened cells, there is indeed upregulation of autotaxin and GDF5, the markers of interval. However, we can observe that the marker upregulation wasn't that strong. It was a mild upregulation of autotaxin and GDF5. So we thought that we are putting Barix1 in an ectopic location in the cartilage. So maybe it needs collaborators to function along with it to induce all the properties robustly of interzone. So to do that, I designed a chimeric protein. That is, I fused Barix1 with VP16. So this VP16 is a trans activation domain. And what this does is, this chimeric protein will go and bind to endogenous Barix binding sites. And because of VP16, all the downstream transcriptional activity would be switched on always, irrespective of presence or absence of a collaborator. And therefore, this serves as a constitutively active version of Barix1. So when I electroporate this, I find again that a very similar phenotype in the embryo altogether. That is, the limbs are short, malformed, and even the distal regions are not formed. But again, you have several segmentations, even here in the pelvic girdle. So if I look into it molecularly, I again see that where you have infection, that region is beautifully flat and has lost collagen expression compared to another internal control, which doesn't have infection, nor flattening, nor there is downregulation of collagen 2. And you can see the same here in safranin O staining, where the central regions are flat cells here, while the other uninfected domains are circular. So I'll just mention here that this is actually the first time we show that a transcription factor was capable of flattening cells in the cartilage unit, as well as downregulating collagen 2. Now, what about the markers for which we got into VP16 number? And that is that with misexpression of Barix1 VP16, the expression of GDF5 and autotaxin along the contour of VP16 infection, it shows a robust expression of GDF5 as well as autotaxin. So therefore, if I put the gain of function experiments together, what we see is that it's sufficient to induce not only the molecular features, but also for the first time, the molecular features associated with the interzone. So this was exciting, and for the first time, hence we were happy, but is Barix1 also necessary? So for that, I initially went for RNAi-mediated knockdown, we went for CRISPR-mediated knockouts in mice, but somehow, unfortunately, both of these techniques failed for us, specifically because of the gene Barix1, which is difficult to work with. I can discuss it later, why we feel that it happened that way. But Barix1 being a transcription factor, we had another way out. So that is making a dominant negative version of this protein. So instead of VP16, I put in engrailed here, which is a repressor domain. So now, again, this chimeric protein will go and bind to endogenous Barx binding sites, but because of engrail, now all the downstream transcriptional activity would be stopped, again, irrespective of presence of stronger or weaker collaborators. And when I electroporate this again through RCS vector, I find that indeed the markers of endozone, they are downregulated. You can see GDF5 and autotaxin downregulated, which are in red, in the regions where there is BARX1 engrailed expression, which is in green. So they are mutually exclusive and the endozone markers are going down. But what about COL2? So being a dominant negative version, we expected collagen 2 to go up and the cells to be no more flat. However, when I look into the serial section, I find that the collagen 2 expression does not turn up in the region infected with BARX1 engrailed. That is a region of endogenous endozone. And neither is there any change in cell morphology. So it fails to revert the cell shape or restore the COL2 expression. So now taking both the gain of function and loss of function experiment together, what we see is that BARX1 is sufficient and necessary for inducing expression of endozone markers, which is GDF5 autotaxin. However, it's only sufficient to induce morphological features of endozone and associated loss of COL2A1 expression, which in itself is an exciting fact. But then,
Professor Shukla has a question.
Okay.
Tathagat?
Yeah.
Professor Shukla has a question.
Yeah.
Arun, please go ahead.
You can summarize it. So I was just wondering, in your VPE 26 and engrailed strategy, the endogenous BARX1 is still there and functional, right? That is not influenced in any way.
Yeah. So, so the thing is that when we electroporate this construct, so it's more of a population effect. So you have more of BARX1 VP16 in much more amount. So the number is excessively high than what is physiologically present in the cells. So by population dynamics, BARX1 VP16, which is electroporated, is in far more numbers than normal BARX1. So it hijacks the endogenous binding domains of BARX1.
Okay. Okay. Thanks.
Okay. So however, the loss of BARX1, as we saw, does not induce restoration of COL2A1 expression, nor does it revert back to morphological changes. So that also brings us to the future prospective question that we are trying to answer in the lab. Initially, the question is very simple. We are trying to understand what leads to the flattening of the cells. So we are, Ankita is actually trying to do it with micromass culture, which mimics the 3D environment and have BARX1 infection to figure out, is it extracellular or is it intracellular factors leading to cell flattening. Secondly, we are invested in trying to figure out a mice model where BARX1 activity would be gone from the very beginning of lymph formation. As you remember here in my slide, endogenous endozones did not upregulate collagen 2 upon infection with BARX1 engrailed, but that could be because it came in later. So if we remove BARX1 early, we might see that it's actually not letting endozone form. So that is the next thing that we are trying to figure out and design the mice of. And the final thing that we will do is that to see if I had shown you that there are exogenous segmentations, but that doesn't necessarily mean that these are functional joints. A functional joint means many more things. It has tendons, ligaments, it has synovial cavity, it moves. So we will see one by one what are the features of an ectopic joint which can be mimicked in these exogenous segmentations which have come out of BARX1. So with that, I also come to the end of the first part of my presentation. The second part is about PRDX1. And before I get into that, I'll just mention that this project is more about identification of PRDX1 as a BMP downstream target. Yes. There's a question from Professor Pradeep Singh. Yes, sir. Are there questions? I can see raised hands.
No, you go ahead.
Okay.
Professor Shukla's hand, I have to lower it.
Okay. So, so when I'm talking about BMP downstream targets, I need to mention here that bone morphogenetic protein, which is both necessary and sufficient for bone formation, somehow we did not know any downstream targets of BMP signaling, which is expressed in the bone. Apart from a single study, which was again from our own lab, where Paritosh and Prem actually analyzed existing microarray data to identify a set of BMP downstream targets. But as you can see from the title itself, they were from existing studies which were actually not done in osteogenic cell lines. So we wanted a better study, de novo, in an osteogenic cell line to identify novel targets of BMP signaling. So for that, we collaborated with Dr. Arun Trivedi from CDRI, and there we used, they used brighter cell lines in proteomics-based approach to identify BMP downstream targets. So the brighter cell lines were actually designed by our own lab. So Dr. Bandyopadhyay and previous students, they have actually already commercialized this cell line and as well as patented this cell line. And there's a lot of features associated with this. I won't get into the details of all of them, but for the present presentation, it's important to note that brighter is a BMP depletable cell line. That is, BMP2 and BMP4 locus are lost here. And again, I'm not getting into the details. When you add 4-hydroxytamoxifen, that leads in these cells to nuclear localization of Cre. So Cre is more of a Caesar-like molecule, or maybe someone who is not initiated to Cre. So this is a recombinase, and this goes and chops off the regions which is being flanked by LOXP. So what happens is with 4-hydroxytamoxifen, BMP2 and BMP4 is excised out of the DNA, and you have a cell line which is bereft of BMP2 and BMP4. So long story short, 4-hydroxytamoxifen equates to low BMP levels. So Dr. Trivedi's group, they cultured the brightest cells in presence and absence of 4-hydroxytamoxifen, ran the lysates on a 2D gel, and identified several differentially regulated genes. But the one which is mentioned here is the one which showed maximum amount of differential expression, and that gene is PRDX1. So PRDX1 is an antioxidant protein which belongs to the generic 2-cis subfamily of peroxyredoxins. Now, the interesting thing is that BMP signaling has not yet been known to work along with an antioxidant protein in terms of bone formation. So this was exciting for us to look into it, what the function of PRDX1 is in terms of bone formation. So before I get into that, I'll just give you an idea of how the signaling systems are in a developing bone, because I'll be needing it to explain some of my data later. So you have in one single unit of bone, 2 proliferative zones. So these cells, they keep on proliferating under the influence of PTHRP. And as they keep on proliferating, they slowly move out of the domain of influence of PTHRP. And as soon as they move out, they start expressing Indian hedgehog and become prehypertrophic. And they do so because initially PTHRP is inhibiting the expression of Indian hedgehog. And therefore you have, if you notice in the cartoon itself, in this loop, there are always 2 domains of prehypertrophic cells and 2 domains of distal proliferative zone. And there is only one central region of hypertrophy. Now I mentioned these 2 units because if I've mentioned earlier that we go for internal controls. So being 2 in unit serves as a good purpose where you can manipulate one of the regions and you can see the result by comparing to the other unmanipulated region, which serves as the internal control. So the first thing I do is to look into the expression of PRDX1, because to have a role in bone formation, it must be expressed in the bone. So when I look into the mRNA expression of PRDX1, I find that it's actually expressed in 2 of the prehypertrophic domains, which maybe you have guessed because I told you earlier. And while it's present in the prehypertrophic domain, it's absent in the central hypertrophic domain. It's better if I give you a zoomed up image where you see the PRDX positive prehypertrophic cells here, while the hypertrophic cells are devoid of PRDX1 expression. Now, I also wanted to ensure that this PRDX expression in the bone is actually under the influence of BMP signaling. So what I do is I use the BMP knockdown construct, which was already used by Paritosh and Prem in their paper, and I electroporate that knockdown construct. This again, I put into RCAS vector, and I electroporate that into developing chicken limb. And there you find again, there is one in A prime, a region, a prehypertrophic region, which is infected. And in A double prime, you have an uninfected prehypertrophic zone serving as the internal control. So in the infected domain, when you look for PRDX1 expression, it's gone. While in the control uninfected domain, you have stable expression of PRDX1. Therefore, BMP signaling is indeed regulating the expression of PRDX1. Now, to understand the role of PRDX1, I decided to knock down PRDX1 and see how the bone forms or how the bone develops. And for that, I designed a knockdown construct and tested its efficacy initially in vivo. And I saw that it's actually effective. That is, in green, you have the knockdown cells where PRDX knockdown construct has gone in. This again has been used through an ArcS vector. So repeatedly, all the electroporations are through ArcS vector. So in green, you have the PRDX knockdown affected cells, and in red, you have PRDX mRNA expression. It's even more visible on the second panel, where you have the infected cells with PRDX knockdown, down-regulating PRDX mRNA expression. So now that I know it's efficient, I electroporated this and looked into, if you remember, PRDX is expressed in the pre-hypertrophic domain. And Indian hedgehog is the marker of pre-hypertrophic domain. So it's logical I look into the expression of Indian hedgehog. And this again, here, as you see, is a knockdown construct electroporation where this first pre-hypertrophic domain is rather heavily infected, whereas the second pre-hypertrophic domain shows lesser infection. And when you look into Indian hedgehog expression, the first pre-hypertrophic domain is actually having perturbed or downregulated Indian hedgehog expression, while the second domain has persistent Indian hedgehog expression, which again was the internal control. So PRDX is abrogating Indian hedgehog expression, or in other words, it's abrogating hypertrophic differentiation when knocked down. But what about the hypertrophic domain? So if you look into this, again, look at the second panel along the pointer, you'll see that the green cells here, which are the PRDX knockdown cells, they do not express collagen 10, which is, in the cartoon you can see, is a marker of hypertrophic cells. So all the cells here do not express collagen 10. However, you know that this is actually a hypertrophic domain and not some other domain, because on the outer edges, you have regions which are less infected by PRDX knockdown construct, and they still show persistent collagen 10 expression. So therefore, PRDX knockdown abrogates both Indian hedgehog expression and collagen 10, as well as in the corollary, it perturbs pre-hypertrophy as well as hypertrophy. So we were interested to see, is the same effect reversed if we overexpress PRDX1? Does it increase expression of Indian hedgehog? Does it increase collagen 10? So for that, I used a full-length PRDX1 construct. I electroporated that. And let me take you through the data. In the control, if you remember, I told you that there are 2 units of pre-hypertrophic cells again, each of which are Indian hedgehog positive. But when I electroporate PRDX1 full-length construct, which is in green here, I find that there is upregulation of Indian hedgehog all across this unit. It makes it a single unit of upregulated Indian hedgehog expression. Now, there is another interesting observation here. If I pseudocolor this Indian hedgehog expression in red and merge it with this PRDX1 misexpression, what I see is that the terminal ends actually do not upregulate Indian hedgehog. It's better if I give you a zoomed in image. That here B is what this box B here shows, that the cells which are initially not to express Indian hedgehog now express Indian hedgehog upon infection of PRDX full-length construct, which is here in green. And the Indian hedgehog expression is in red, all these cells. However, despite upregulation of PRDX full-length construct, there is no upregulation of Indian hedgehog here in the terminal ends. So I'll come back to it and what we think about why this is happening again. But before that, we will look into what happens to hypertrophy. So here you can see a bone, which again is very nicely, half of it is infected. That again serves as, in the yellow asterisk is the test region, while on the white asterisk is the internal control region, which doesn't have infection. So in this infected domain of PRDX full-length construct, you see there is downregulation of collagen 10. Whereas the uninfected domain, the internal control has normal expression of collagen 10. Therefore, PRDX1 misexpression as well as knockdown both impairs hypertrophy. In fact, when we looked into a skeletal prep of PRDX1 overexpression, we saw that it actually mimics what was found in mice. So here you see that the bones are stubby and with lesser ossification. The same was observed by Andrea Broadkamp's group much earlier when they actually overexpressed Indian hedgehog in mice. You see the bones are stubby and there is uniform domain of Indian hedgehog expression. So if I put this, what we observed, into a model to explain what we see, so in the pre-hypertrophic domain, you have in blue the Indian hedgehog expressing cells, wherein also you have pre-hypertrophic cells expressing PRDX1. The central region is collagen 10 positive, and in the terminal ends, you have PTHRK and its domain of influence. However, if you knock down PRDX1, what happens is it downregulates Indian hedgehog expression. Now, as it downregulates Indian hedgehog expression, it actually perturbs prehypertrophic differentiation, which in turn stops every kind of progress into the hypertrophic differentiation. Therefore, with PRDX knockdown, you get abrogation of both hypertrophy and prehypertrophy. However, when we overexpress PRDX1, which is in red here, you find that it upregulates Indian hedgehog. That is, it sustains the cells in their prehypertrophic state. And now, therefore, because they're sustained in their prehypertrophic state, it cannot get into hypertrophic differentiation. So here, though Indian hedgehog is upregulated, there is no upregulation of collagen 10. And the reason what we think that there is no upregulation in the terminal domain of Indian hedgehog, despite upregulation of PRDX1, is that it's still under the influence of PTHRP's downregulation, which doesn't let Indian hedgehog come up. So this is the model that we think is working, that PRDX1 downstream of BMP signaling is regulating Indian hedgehog expression. But this is what we don't know. How is it regulating? So we have an idea which we are working on, that because PRDX expression actually complements what is observed in mice bones of the prevalence of ROS abundance in developing bone. So they see, Morita et al. Had seen that hypertrophic cells are rich in reactive oxygen species, that is ROS. And it goes down as you move away from hypertrophic domain. So PRDX's expression in the prehypertrophic domain might be ensuring this drop in ROS, which in turn might be regulating expression of Indian hedgehog. Because there are instances where ROS has been regulating expression of sonic hedgehog, which is a family protein. And with this, I also come to the end of this part where I'll be summarizing that BMP signaling promotes the expression of PRDX1, and PRDX1 regulates expression of Indian hedgehog and therefore sustains the hypertrophic state, presumably by regulating the ROS abundance. And we published this story with Dr. Trivedi's group from CBRI. And with this, I also come to the end of it, and I get into the DPYSL3 story, which is a BMP downstream molecule. Now, I already introduced you to the paper which we had from our lab, that is Paritoshi's paper, where microRNA meta-analysis actually revealed to us a set of genes which are downstream of BMP signaling. So DPYSL3 was one such gene there. Just to give you an early intro into it, you can see a wild type mice which has both BMP2 and BMP4. There is a strong expression in blue of DPYSL3. However, in a BMP2/4 knockout mice, you see that there is downregulation of DPYSL3. So it is downstream of BMP signaling. But that's interesting because DPYSL3 is actually quite a well-studied molecule in the context of neural development, where growth hormone dynamics are known to be regulated by DPYSL3, which is the dihydropyrimidinase-like 3 protein. And it's also known as the CRMP4, which is the CRMP stands for collapsing response mediator. So you can understand it's mainly related to neural development. What was also known about this gene, that it's known to interact with regulators of exocytosis and endocytosis in worms. Now, this paper was actually interesting, and that is why I'll be coming to it later why it's interesting. But this led Paritosh and Prem to look into cell secretion, because exocytosis and endocytosis is related to secretion in worms. So Prem and Paritosh actually transected the DF1 cells with the Gaussian luciferase expressing construct. As you can see, and it's one of the luciferase which is secreted readily onto the supernatant. So you can see in the untreated cells, there is the expression of Gaussian luciferase, or the levels of Gaussian luciferase in the supernatant. It is equal to what was seen when was observed with a control microRNA, that is LAGZ microRNA. However, when DPYSL3 microRNA was introduced, so this knockdown actually downregulated the amount, or rather abrogated the secretion of Gaussian luciferase onto the supernatant. However, this could be rescued with a rad DPYSL3. Now, rad DPYSL3 actually escapes the downregulation by DPYSL3 microRNA because this is against the chicken mRNA. So DF1 is a chicken cell line. So rad DPYSL3 could rescue it back to its normal levels. And even more exciting was the fact that with rad DPYSL3 transfected alone in the DF1 cells, you see very high levels of Gaussian luciferase into the supernatant. Now, I come back to the point why this is interesting, because when you imagine bone, bone is 70%, an adult bone is 70% mineral phase, which is interesting because the other part, which is 30% organic phase, only has 2% of cells. So the cellular content is only 2% here. That means the rest of it in the collagen matrix, which is 97% of organic phase, is actually secreted by the cells onto the extracellular matrix. So if DPYSL3 is regulating cell secretion, then it would adversely affect formation of bone if this knockdown was done in the bone. That was our prediction. But before that, I wanted to understand what exactly is affected in the cell by DPYSL3. So for that, I worked in collaboration with Dr. Mahek Sharma from IIT Mohali. And we know that secretion happens through Golgi, or in some cases through the help of the cytoskeleton of the cells through which the exocytotic and endocytotic vesicles are transported. So we looked into both of them. So here initially you see a LAGZ control microRNA, which was again previously used by them. I see that there is proper Golgi organization in the same place, and it's not all over the place if it was perturbed, which is in that case. And the actin cytoskeleton here you can see is pretty normal with stress fibers, the central stress fibers going from one end to the other. It's better visible if I put on the video where you find the stress fibers going all across the cells beautifully. However, if I look into DPYSL3 knockdown cells, again, the Golgi, interestingly, is quite unperturbed. It's still together. But if you look into the actin stress fibers, you find that they are gone. The central stress fibers are gone from the cells. Now that's exciting. And to know if it's actually conserved in mouse cell lines and BMSCs as well, I looked into it, that here in the central region, you find the DPYSL3 knockdown cell as in green, and the other cells which are not affected by the knockdown are not expressing GFP. So this knockdown construct has GFP. And this knockdown construct actually shows loss of stress fibers again. And you can see the stress fibers are actually present in the uninfected cells here, which again you can term as internal controls. So stress fibers are gone when DPYSL3 knockdown happens. Now this phenotype is reminiscent of what we know if we do a quick literature survey of RhoA inhibition. So RhoA actually inhibition abrogates central stress fibers and keeps up the cortical stress fibers, which is also seen in this case. Stress fibers are present, while the central stress fibers are gone. Now, if that is so, is there any relation between DPYSL3 and RhoA? So literature reveals that actually DPYSL3 and RhoA are known to interact with each other. So Alabab and the colleagues have found that the first 128 residue of the N-terminal domain of DPYSL3 is enough to interact with RhoA. And actually they cloned this 128 residue and used it as a competitive inhibitor of DPYSL3 and RhoA interaction. So with C4RIP, which is C4 is for CRNP4, R for RhoA, IP for interaction inhibitor protein. So when you have C4RIP, any function mediated because of DPYSL3 and RhoA interaction would be thwarted. So they gifted this construct to us, generous gift from the lab, and I introduced this C4RIP construct into the cells. And indeed, the stress fibers are not gone in this case, but they are malformed. You can observe them better if you look into the control cell where there is linear taut stress fibers here, while the stress fibers here are with perturbed morphology. It's not linear, it's not taut from one end to the other, whereas the cortical stress fiber remains. So what we see is that DPYSL3 knockdown is actually abrogating stress fibers, and when we inhibit the interaction between RhoA and DPYSL3 through C4RIP, which is a competitive inhibitor, we see similar phenotype of loss of morphology of the stress fibers. Now, if this is so, if you remember, we were curious to know if DPYSL3 could induce any kind of problem in bone formation in vivo. So first we'll look into what is the effect of DPYSL3 in vivo. So when I introduced DPYSL3 knockdown in developing chicken limb, initially, morphologically, there is no effect whatsoever. If we compare this, this is very normal compared to what was in the case of BARX1 or PRDX1. This is a perfectly normal limb. However, the femoral region lacks ossification. As you can see in the contralateral element, there is a very clear red domain of ossification. So when I take sections of these tissue, I find is that the infected domain actually does not have collagen 10 secreted onto the extracellular matrix, whereas the domain, which again serves as an internal control, where there is less infection, there's a lot of collagen 10 secreted out onto the extracellular matrix. Therefore, as we expected, DPYSL3 is actually abrogating the secretion of collagen 10, and therefore it's abrogating ossification altogether. But if what we observed is true in vitro, that it's because of Roe's interaction, then if we introduce C4RIP in this developing cartilage, we will have a similar effect. And I introduce a C4RIP again through RCAS construct. And when I electroporate that, actually there is no collagen 10 expression in the cartilaginous unit. And now I needed a contralateral control because there is no expression here at all. So this mimics what we see in case of DPYSL3 knockdown. So what we can conclude is that DPYSL3-Roe interaction is important for regulating proper cytoskeletal development, as well as maintaining collagen 10 secretion during ossification. And with that, actually, I come to the final summary, where it's not a summary, rather a presentation, a graphical presentation of what I had found, that in induction of Endozone, we found that BARX1 is a sufficient molecule to induce all the features. It's not necessary, but only for the morphological features and the collagen 2 expression downregulation. Otherwise, it's both sufficient and necessary for the markers. What we see in case of prehypertrophic differentiation is PRDX1 sustains the expression of an inhibitor and therefore helps in prehypertrophic state maintenance. And finally, we see that DPYSL3, which is a downstream molecule of BMP signaling, is actually regulating cytoskeletal dynamics with the help of Roe interaction and thereby regulating the ossification in a developing bone. So I'll now come to the acknowledgement slides. So yes, I have prepared a lot, but now I'm messed up. Like both Dr. Bandyopadhyay and Dr. Sen have been, I really cannot put in words how they have been as PIs. Like it's more, I won't be overstating if I say that they have been more like semi-parents, especially with a lab like us. We have been extremely unruly and difficult to contain, I guess, but they have been incredible with the way they have guided us, especially me. I knew nothing of it, and they have put in a lot of effort to make me understand most of everything. And there's no clear way in which I can actually thank them. If the only way I think is if I can do really good as a human or professionally one day, and I can make them proud of the fact that they took the risk of taking me into their lab. So thanks a lot. I really don't have the right words to put in right now. So then the BSB faculty members, I guess this department is fantastic as a family. You would be surprised to know that Professor Sinha, he actually spent more than a week to change my abstract, which you found in the mail. He dotted the I's and crossed the T's and took 1 week to make it perfect, and so that I can mail it out to every other person in the department. You have faculties who come up, who you meet in the corridor, and they put you through the best of their inputs, like how you can better your talks, how the projects are going, and they even explain their own data to you. Professor Shukla, Shankar said they would be inquiring about football, how the research is going. It's incredible department. I'd like to thank all the faculty members actually. It's a brilliant department that we are in. And the BSB office staff, you don't have to think anything about any other stuff. If you just can concentrate on your work, and they will be doing all the paperwork. And it's incredible how they do it. Not so many people around. And then I would like to thank all my collaborators who actually have been very helpful, and especially Professor Mahek Sharma, because she actually hosted me in IIT Mohali for 2 months, and it was a very exciting experience for me to work in a cell biology lab. I learned a lot. And then my past members of lab who have been like the real leaders in making me understand everything, and without them I wouldn't have understood much of anything of biology, I guess. And then my entire lab, in no order of preferences, but it's without them, and they know how much I miss them and how bad it is that I have to give this defense online. But I hope we can meet again one day, and we'll have a lot to enjoy about. In Barkha, without her, the lab would not function at all. And Naresh Bhaiya and Deepak Bhaiya have been impeccable in their duties of handling all the mice and chicken work. my friends Shubhabrata and Madhusri, who have been the Bengali connects in my department, in my stay in IIT, my batchmates here as well, and all my other friends, football team and Holly football team, which have been going more regularly than even if I still have. I've been here all the days in my IIT stay, and my travel funding agencies who have actually funded my travels to the conferences. Here, actually, maybe I should put up an image of Dr. Bandyopadhyay himself, because he funded one of the travels himself. So, but yeah, I thought it would be tricky to have this picture here. And finally, IIT Kanpur and MHRD for stay and scholarship. And I don't know how I forgot, but Kanpur city itself, it's a fantastic city with a fantastic herd. I really would like to get back there one day, and I really, really miss Kanpur. And finally, my family, and most of them are all online. My grandparents, my ma, baba, dadu, dida, mamu, mami, bhai, all of them are together in each of their systems. And it's exciting to give actually the defense and meet them immediately, which is the only good thing of this online stuff that I'm doing. And that's it. So thank you everyone for listening, and thanks everyone for coming, the committee and all who are present in this open seminar. Thank you for coming.
Okay, Tathagata, thank you for the talk. Now we will open up the floor for questions. First, I will go to Dr. Deepak Modi. Deepak, if you have any questions.
Yeah, Tathagata, that was a wonderful presentation. Am I audible?
Yeah,
Yes.
Yeah, so that was a wonderful presentation, and it's great seeing you again after the NSTV meeting. And I'm sure you'll agree with me that Bandyopadhyay lab has transformed the electrical engineer into a into a biologist. So there's a very strong expression signal, and I really love that. Amitavu, very wonderful, very, very nicely done. I'm very impressed, and I've gone through the questions and answers that you provided, and I don't think I really have any further questions. I was just curious in the Barracks 1 story that, could you still elaborate on whether you think these are still cell autonomous effects, or you cannot convincingly talk about those two?
I really cannot clearly say if it is non-autonomous, but what we really think it's actually cell autonomous. If you're talking about collagen 2 downregulation, which I believe is completely cell autonomous, because it's there are a lot of data. I had 3 different sections of both BARX1 and VP16, which makes it to 6 sections. All of them shows conspicuous downregulation of collagen 2 in each and every cells which are infected. I have a slide, I can show you that again, but okay, I guess. So that is very cell specific. Now, but if you're talking about cell flattening, that we are not very sure of, because there might be several factors. The first thing is that collagen is in the extracellular matrix, providing strength to the matrix. Now, we really don't know, is it the cell flattening, is it because that there is loss of collagen and then it's actually squishing the cells? Or is it actually because of some intracellular molecule, like something like GPU-ISL3, which is actually regulating the cell shape change and accentuating? It might be both together. So we will, in the micromass culture, we will be actually manipulating these stuff, like just remove one of the stuff and see what the other can do. Just remove collagen 2 maybe if possible, and see if only the forces around the extracellular factors can induce the cell flattening. So right now, I guess it would be very difficult to say if it is cell non-autonomous, but what we have observed, most of the features are very cell autonomous. It's only in the domain of expression of BARX1 you see upregulation of the markers or downregulation of collagen 2, and as of now, the cell flattening as well.
Okay, I think that's convincing. And the second story, if you can go to the slide where you could show that the PMP. Yes.
The PRDX1.
Yes.
Here.
The one before that.
This is.
Yeah, the one before that.
One before this?
Yes. And one more. Yeah, so in the pre-hypertrophy and the hypertrophy zones, so you could show that despite overexpression of Indian hedgehog, the distal zones could not be, did not undergo pre-hypertrophy, right? So one of the reasons that you suggested was that it was because of the inhibitory effects of PTHP. is what you
Yes,
suggest,
it is.
right?
Yeah.
I was curious that is it just that could it be just PTHP, or that something from the central region is not sufficiently going on to the sides? I mean, could there be an overlay of a third factor which is not yet identified?
Yeah, that actually can be a possibility. Though, like, we don't know of a molecule as such going through, having an effect all through the cartilage element. But what actually can be, as you asked this question, I have a feeling, what can be a particular kind of regulation is some exact threshold of ROS, which might be present in that region, which is not present when you travel finally to the very end of the cartilage unit. And therefore, it might actually regulate the Indian hydrog expression. That can actually be true. Maybe it's there's a bracket of exact abundance of ROS, which is okay for Indian hydrog expression, which is either maybe too low away from the hypertrophic domain and too high in the hypertrophic domain. Yeah, that is a possibility. So we don't know that, but we don't know of a molecule as such also. So PTHRP seems to be the most knowledgeable
so that could
guess.
be an alternate hypothesis.
But that definitely could be a way of regulating.
And I missed this. How does BMP overlay here? So if we were to look at BMP expression in this region, how does it relate to?
I don't actually have a BMP expression data here. So BMP is anyway secreted on, ligands are secreted. So more importantly, if I just give you an information, so I don't have this in my thesis or presentation. So Shuchi from our lab, she's working on it. So we are looking into the actual expression domain of BMP signaling, which is like looking into phosphosmad 158, which is the readout of BMP signaling, rather than looking into the ligands per se. And what she found is that we have yet published that it's actually expressed strongly in the hypertrophic domain itself, which is exciting. So when we publish that, we will be connecting with this data of PRDX1, which is exciting that BMP phosphosmad data is actually in, with what we see is actually in the pre-hypertrophic domain. So it could be directly regulating PRDX1 in that domain itself.
Right. So exactly that was my question. That is BMP. So is there again like the threshold of BMP? Because since these are morphogen gradients, do these exactly overlap with each other, or are this again thresholds which is now creating this segmentation like pattern?
Yeah, that's. I guess I really don't have an answer right now because we haven't looked into the gradients of BMP signaling levels or as such. But that again could be a reason why it is so. But even in the, if I just can put it that way, when we are looking into phosphosmad expression, which is which she finds in the prehypertrophic domain, we, if I remember correctly, we do not see any such diminishing gradient. It's more of a block of expression in the prehypertrophic domain. It's more of a on-off band pass filter kind of an expression where you have sudden expression in the prehypertrophic domain and then it goes away of phosphosmad positive cells.
Okay, great. Thank you. I'm good now.
Thank you.
Okay. So Deepak, if you have no further question, I will go to Dr. Shiva. Shiva, do you have any question?
I must congratulate you, okay, as well as Amitabha for transforming an electronics engineer into a biologist. Okay. So our hearty congratulations to you. So I have a couple of questions. So I know very little about this area, but I also know very little about this autotaxin. Okay, because since we are working in the cancer proliferation rates and everything. What we understood is that, okay, since autotaxin plays a very major role. So for our understanding purpose, okay, suppose if I want to, okay, is there any commercial FDA approved inhibitors available for this autotaxin?
I honestly have not looked into inhibitors of autotaxin, but we can look into that. So autotaxin is one molecule which we haven't investigated as such. As you can see that mostly it has been with GDF5 because also the enhancer of GDF5 was characterized, and so it was more of a GDF5 bias project in that sense. So we haven't looked into inhibitors of autotaxin, but you just want to know if there is any inhibitor which we can be using, or in terms of like what effect it would be inducing in the interzone?
No, it is okay. I'm just looking for the commercial or FDA approved inhibitors.
Oh, okay. Sorry for that. Anyone else might have any information they can share, but I really don't have an idea of any FDA approved autotaxin inhibitor as such.
Okay, because what I found it is that okay, we are thinking of using some exosome or two, okay, to inhibit those things. That's what I was thinking. Okay, that's okay. We'll look into it. The next one is that okay, in your slide number 24, I find okay some directional growth in the cells. Okay, I think 24 or 25. I think 24.
Is it this one?
No, this 39. Yes, yeah, this one. Yes, yeah. So I see. Okay, that's a directional growth, right? Okay, in the cells, right? The top left one, the green one.
This one?
Here, the square which is okay.
This one?
Yeah.
Okay.
So is there any particular reason behind it?
Directional, you mean in this direction?
In this direction, yes, yeah.
Yeah, so there are two possibilities and there are two information that I would like to share because you're asking about this. So in general, what happens is this RKS bar X1, apart from infecting its replication competence, so actually it has both horizontal spread as well as vertical spread, that it can infect new cells as well as it goes through clonal population of cells as well, because it gets into the DNA and gets incorporated there because of viral machinery. So there is a possibility that these cells are randomly infected without any such directionality, but there's also a possibility that these are clonal population, which at times there are, like we know there are studies which say that the cartilage cells, they divide in one axis and then they turn themselves up in a 90-degree, and then they align themselves in the same line as each other. That is, they would be different, they would be dividing in these axes in my pointer as I show, and then they turn around and be juxtaposed to each other. So these could be either of the two, but I think it's more of random infections here because these are replication competent virus. So any cells which are secreting that can finally infect any other cells, even though they are not related to each
other. So Tathagata, here was my question. Am I audible?
Yeah.
So here was my question, that these cells which are randomly infected with your construct still do not show collagen expression, right?
Yeah, they do not show collagen expression.
So I was just curious, whether you still want to call it as autonomous. There's several areas which is very high. So one, as I agree with you that it could be certain threshold required to be reached, because I see that in most of your sections that not everything that's green is also collagen positive. So I feel, it's probably dual, that, some parts of it are being done by the molecule and some of it is still cell non-autonomous mechanism, which is just fine.
Yeah. So actually what I was talking about is this additional slide where you can see there are just some random cells here which are expressing collagen 2. And again, you see those random cells actually down-regulating collagen 2. But I understand your point because there is one such spot where there is down-regulation of collagen, but there is no collagen yet. It could also be that a very rare possibility could be that it's actually very early infection. And by the time we detect that and all the proteins, so what we detect here is mainly the coat proteins of the virus. So maybe it's not expressed, but maybe if the call2 downregulation is very fast, like it's an immediate downregulation of collagen 2 with expression of RH, then maybe there could be that. But I think I agree with you that there is a mixture of both. There is cell autonomous. What we really feel it's autonomous right now, but it could be very well coming in from some other factors together, which we will dissect out in the in vitro study.
Perfect.
Yeah, so coming back to that, I don't think it's directional per se, but there is actually no way of rightly knowing if it is directional infection or not.
Okay, fine. That's all from my end.
Okay. Arun, you have any question?
Yes, I have just two quick questions. So Tathagata, It was a wonderful presentation, very clear. And this is, so the first question I'm sure that I have asked you multiple times, and I have asked this to almost everybody from your lab. So you mentioned in the beginning that oftentimes you see that different embryos, they show different morphological features or different patterns of staining. So is there a way to quantify these in situ data that you generate?
In terms of, you're talking about simple in situ or in purview of this Barexon project itself? Like,
so in general, the staining that you are showing.
Oh, yeah, I guess, yeah, we can quantify
I know
that.
Deepak is smiling because, perhaps this is well acceptable in the field, but for us, if we don't show the quantification, forget about publishing it.
No, so we have, like when we do quantify if there is ambiguous expression where we are not very sure of it. But right now, like even in the PRDX paper, we have shown the clear domains of expression in which was accepted. And so, but we can quantify, definitely we can quantify. But then if it is very obvious, which is like observably clear, like say, for example, here, if you can see that the hockey stick region, which is infected, shows very clear down regulation. So we have not quantified those things as of now, but what we are going to quantify more importantly is how much and how often the self-flattening happens in the in vitro setup. This would be easier to do as well, because here, as I was talking to Professor Modi, that it might be because of several other factors and still non-autonomous and because of the region that they are in. So taking a section and quantifying the features might not be perfect at this point of time. We might be missing out on effects coming from adjacent cells or the adjacent cells which are flattened, etc. So, but the simple answer is we can definitely quantify the in situs, but we have
So in other words, for example, if you did the same experiment in 5 different animals or embryos, wherever you're doing it, you will see exactly the same pattern, right? Or how much variation there is across different samples, let's say.
So that depends on the experiment. Say for like, as you can see, the morphological repetitive phenotype in these embryos are always the same. Like all the embryos which you find in infection of say C-Barx1 or especially VP16 Barx1, you see in case of VP16, it's gone. The limb is completely gone almost. So that is repeated in every embryo that you harvest. So that's morphological aspect of it.
This is for my own curiosity. This is not a criticism. And in the final story where you talked about this comparative inhibitor of RhoA DPYSL interaction. So this inhibitor construct was designed specifically for RhoA and DPYSL3 interaction. Is it possible that it can inhibit the interaction of RhoA with other things as well? Because as you know, RhoA interacts with so many things in the cells.
Yeah. So actually DPYSL3
so Amitabh, you mean the first question was not valid? Yeah, go ahead.
I'll come back to the first question later.
Yeah, go ahead.
So DPYSL3 is definitely is a family of proteins. So there are other DPYSL3s as well. So they might have very similar domains. But what we see is that if we put in rad DPYSL3, then we can actually rescue this phenotype. So that, which is not listed here, we have just one in for that, so it's not listed here. So I think it's specific because with DPYSL3 we can rescue that. So I have the data for DPYSL3 knockdown, not after C4RIP knockdown in the presentations right now. But it is that because DPYSL3 is able to rescue that, so there is a fair possibility that it's actually specific to DPYSL3 and RhoA interaction.
No, and actually, I would interrupt once. I think Arun's question is something different. What Arun is asking is that how is it known that this particular fragment only disrupts RhoA's interaction with DPYSL3 and not any other partner? Like RhoA interacts with so many other things, and it is such a critical player in many aspects of cell biology. So candidates which are not in our radar, if they are interrupted, then also you can see this phenotype. But I think this C4-RIP is a fragment of DPYSL3 protein, right? And not of ROA.
No, it's a DPYSL3 N-terminal domain.
Yeah. But anyway, you should answer it from that perspective. I do not think you have done a rescue with rat DPYSL3.
No. So in this context, I haven't done the rat DPYSL3 rescue. So that is what I was talking about. So this we don't know, but they identified C4, but actually even that doesn't answer your question. So yeah, that possibility remains, because the way they identified it was that they pulled down ROA and looked into all the proteins which were interacting with them, and then they looked with antibody, and they found that it's DPYSL3 alone which is interacting with, and it is through 128 residue that they put in, and then they pull it down and they find that DPYSL3 is not. But they haven't looked into other molecules if there is any trouble with their interaction, or if it is that it's occluding other domains of ROA which might be important for other molecules interaction. So yeah, that I have not looked into. So that I don't know. Especially if a binding of C4-RIP is actually troubling the conformation or something which is occluding other interactive partners.
Fair enough.
So,
good. That's all from my side.
Arun, about that quantitation part, I try to clarify one aspect, is that when you move to mouse, there it is more amenable to quantification, because the genotype of the cells is uniform throughout the tissue. Okay, here, because it is sporadic, patchy in nature, therefore it is a whole lot more difficult to quantify, number one. And number two is because we generally show one-to-one correlation between cell that has undergone genetic modification and the neighboring cell that has not undergone genetic modification. Therefore, that is accepted in the field as proof enough. It is kind of similar to what you see in Drosophila literature as well, because in Drosophila also you do this patchy expression of transgenes and you show correlation within the patches. But if you, on the other hand, if you see data from our lab or any other labs in mouse, say for example, we are quantifying bone, okay? So then we will do the microCT and then we will quantify it in every aspect. Or if we are doing cell culture based experiment, there we do quantification, pretty rigorous quantification, but not necessarily in the chick in vivo experiments. That has so far not been the practice. I'm sure it would not hurt, but it will just be a whole lot more difficult.
No, I completely understand that if the phenotype is day and night, black and white, it's very clear. But let's say you are trying to make a difference between 70% and 30%. Will that be possible?
No, so that will, those kind of experiments are not advised in chick.
Okay.
Like those kind of subtle differences are not advised at all in chick.
Okay.
So this is in general the developmental biologists who always believe that if a phenotype which has to be quantified is not a phenotype. It has to be just a yes-no kind of a condition. So this is one difference, because I work in both the domains and I can tell you that in absolute cell biology domain, everything has to be quantifiable. But when it comes to developmental phenotypes, it has to be like a yes-no condition. And you have to observe it in more than 70 or 90% of times. So I think that's one reason why most of this data is not quantified, but I think that that's how the community is.
Nitin, talking of cell biology.
Yeah, I was just about to ask the question that Arun just asked about quantification. Like, every time we see images, we immediately think about how to quantify this.
Yeah. Next time I will get a cell biology thesis.
Yeah, so I was curious to know, like, before my questions, like, Tathagata, amazing presentation and excellent words. So congratulations to you.
Thank you.
And so how would you, if you want to quantify it in cell culture or cell lines, in what line we should go about this? That's my first thing I would like to ask. Like, if you isolate these cells, will they still be, they might lose the context, or they will still preserve these protein expression levels as such?
Like I would have loved, there are actually, we are trying to collaborate with a lab in IGIB where we are trying to make the tissue transparent. And once we make the tissue transparent, when we know which are the cells which are expressing BARX1 maybe, then we can look into the cell morphology much more easier, in a much more easier way if we say stain it with colloidin and see the outer cell boundaries. But right now we do not have that. So the other way around, what we are trying to think of is when I'm talking about, say, micro mass culture, so those pellets of cells, we can image all of them together, right? And they are easier to be permeabilized or made into transparent pellets. So there we can actually look at cells, or the number of cells which are infected. Say I have 100 cells infected, and then because I have the entire 3D aspect together, which is not possible right now because I'm taking sections. So it's always, I'm out of context with all the other sections, which might actually be having better flattening because it's coming from both ways. It's extracellular factors and intracellular factors. So my idea is that when we have the cell cultures ready and when we have BARX1 infection, we will actually have the entire 3D imaging of the pellets and see how the number of cells undergoing flattening correlates with number of infections as well as the radius of the flattening with the radius of infection. So that is the plan right now, and it will be very good if we can actually permeabilize the cell and make it transparent and look into the limb part specifically and see how it is. And then we can actually quantify the endogenous BARX1 cell flattening as well, which is also not known. So that would be both way quantification once we can make the tissue transparent easily in the chicken.
Yeah. Great. So I have some naive questions here. So, like in your slide number 11, you show the BARX1 level expression levels in different stages of the, right? So this is not your work, and this is a reference that you're putting, I believe.
Yeah.
But you might have observed something similar, of course. And my question here is like, as as during these developmental course, the course of development, you still see BARX1 very much localized towards that endozone, and you don't see, how it is maintained over there. It's like it is not still intact over there, right? The cells are not migrating or proliferating or going in random directions, and does BARX1 has a role to keep the endozone cells very much intact over there?
Okay, so yes, they persist. I mean, the expression persists till HH44, which is the report. So in your question that whether the cells, I guess what we are trying to ask is it that the BARX1 positive cells, which is the endozone cells, they remain not only BARX1 positive, but it's the same cell in the same location for as long as the differentiation process is going on. So I think what we believe is that BARX1's expression is more about the regulation, as I told you about in the couple of slides later, that we think that it's actually regulated by RA and FGF's gradient. So instead of actually cells being specified that they are expressing BARX1 only, it's more about RA and FGF gradient deciding where the BARX1 expression would come up. So it might be a cell which is expressing BARX1 at this point. If it moves away, it goes to some other gradient point of RA and FGF gradient module, and then they lose BARX1 expression. And it's a new cell which has come up this region is now expressing BARX1. So what that does is, as such, it doesn't let, because I told you that interzone is actually important to have these very specific ratios of limb, which is important. So by any chance, if we make the cell specific to express BARX1, it might be that moving around during development or something would lead to morphological deformities in forming bones. It's actually better to regulate it in terms of a gradient, which will always make a ratio, a portion of the cell BARX1 positive. So I think it's more about this gradient determining the cells which will be BARX1 positive, and then it remains as such. So that is what we think is going on.
Okay. And one last question. So towards the last part of the talk where you're knocking down DPYSL3 and you see the stress fibers, right? Gone. You see the stress fibers gone. And similar is the effect with Rho A depletion, I believe. So the question here is like, as others also asked, like Rho has many other binding partners and it can have the same effect, right? So to understand this, can we just, do this? Like you have a Rho depletion and you rescue that with DPSY-SL3, or you have a DPY-SL3 knockdown and you rescue that with Rho overexpression. Would that save your phenotype or rescue your phenotype?
I think because interaction of both are important. I think if you knock one down, the other won't be able to revive that even if you overexpress, because it's the interaction which is important for this, what we believe. So, but it might be that, yeah, so that's a simple answer, that I think because it's interaction, so both have to be present in say sufficient quantity, both have to be present in say levels X, and then only you have the phenotype. If you remove DPOSL3, putting more of Rho A won't lead to more interaction with DPOSL3 because that is the limiting reagent there. And in the other way around, it would be Rho A going down would again have normal levels of DPOSL3. That too won't lead to that, because that experiment itself shows that here there is no perturbation in Rho A, but DPOSL3 knockdown is leading to the phenotype. And in this case, there is only inhibition of Rho A. The DPOSL3, if here it's all right. So you need, it's more of an AND gate where you need both of them together, and then you have an output. It's either one would not lead to this output. So I think we need both of them together, and then we have the proper stress fibers.
Okay. So there's a selfish question here out of the context. So how about microtubules? Don't see any effect of microtubules.
Oh, so I stained for microtubules. So there is actually no effect on microtubule. Initially, we thought that because it's we are staining with PFA fixation, maybe we are not able to look into it. So then we use methanol fixation. So we removed all the noise. We could only see simple microtubules, but there were no such problems in microtubule, or even because Mike Sharma, her lab is invested in lysosomal pathways, etc. So I also stained for LAMP proteins, etc. So there were not much localization defects on those as well. So the only thing that we could find difference, and repeatedly in several other cells, is that the stress fibers are gone. So the microtubules remain pretty much perfect under DPY-SL3 knockdown.
Okay.
Yeah.
Okay, so I'm done. And again, once again, congratulations to you. I just, if I'm allowed to, I just want to hear a little bit. Maybe everyone here wants to hear a little bit about your transition from electrical engineer to, the biologist that you are, which is amazing transition, which is already here. So just the biggest challenge that you had, can you quickly share with us?
I mean, it might sound funny, but actually initially the biggest trouble I keep on remembering is that I did not understand what genes are, because at times I would think that it's protein, and at times they say that it's DNA. So initially I would be completely messed when they say knockout. Is it like it's squishing the protein, or is it like something to do with DNA? So that was very confusing initially. But then after knowing that, everything was much easier.
I could relate to that very well. So go on.
So yeah, after that it was, but then I would really say that again, like Dr. Bandyopadhyay and both Dr. Sen, they invested so much time in making sure that I understand those from the point of view of engineering. Like very rarely we would be talking in terms. I remember I actually have the answer sheets which I've kept where Dr. Sen, she was taking an exam, and I had written something and it was actually not possible. I told them that I would be putting up an artificial protein or something really crazy. And she still gave me number, and she told me it's practically impossible, but logically it's perfect. So those things actually also made me kind of more confident of what I'm thinking. If I was given 0, I would have thought that I've completely thought wrong. So knowing that I thought right, but I did not know the details, made it easier. So I knew exactly what to focus on. So I guess the way they trained me was impeccable. It was scary because at that point of time, there was no one as such in the department as well. So it was always fun. But sadly, now I would be a biologist, so I cannot use that to my benefit now.
Sure. Thank you.
Okay, Pradeep, I know that you had some questions which I could not attend to earlier. Please go ahead. Pradeep, you are muted. While Pradeep is unmuting himself, anyone else from the gathering has any question, please? Yeah.
Can I ask?
Yes,
Amitabh?
please, Nitin. Nitin, go ahead.
Tathagat, wonderful talk. Really nice. Nice work. Congratulations to you and Amitabh. So my question is related to, I think you already touched upon how BarX1, how the zone is decided by the two gradients, the ratio of the two, the RA and FGF. But can you comment on how exactly the gradient results in the expression of BarX1 in the middle?
Yeah, so that is Upendra's thesis. Upendra will give you in elaborate detail. We have some data that suggests that you need a critically low threshold of both. We have done experiments that suggest that very high RA or very high FGF by it themselves is not sufficient. You need to have both, but at a very low concentration.
Okay.
And molecularly, how that translates, we still don't understand.
So again, I mean, it's part of Upendra's work, but we are trying to understand the enhancer elements of BarX1 as well, to know that if there are downstream targets of FGF and RA, which might be playing a role in the enhancer element, and see if there may be some chromatin level regulation as well out there in this.
But that would, I mean, since you, as you show in the figure, and I can also imagine that the gradients will change slowly. So it will not be a sudden jump from one place to the other. So how does the BarX1 get localized to such a narrow region? There must be some other feedback loops that further restricted once it, so I look forward to seeing more in Upendra's work, maybe.
Yeah, like, I'll tell you one little experiment that Upendra has done. So he guessed the zone of BarX1 expression because see, you have to understand that all the manipulations are being done at a limb bud that is much earlier than where BarX1 is expressed, or you get to see actual joints. So he put another RA bead there, and then you get a new BarX1 expression little bit away from the RA bead. So, and there is a reciprocal regulation. RA inhibits FGF and FGF inhibits RA. So that loop is also operational. So it is a rather complicated story, like another student, Ankita, she was all set when the lockdown came. We now have a cell line. It would be a fantastic thing if we can recapitulate this flattening in the cell line. If we can do that, then we will be able to analyze the transcriptional regulation of BarX1 in much finer detail than what we can in vivo. And then once we have the detail, then we can try and reflect back in vivo. So, Ankita has another 15 years to complete her thesis. So she will tell you more about it.
Okay. Well, thanks. Thanks, Tathagata. That was really wonderful talk. Again.
Thank you. Thank you so much.
Anyone else? Shankar? You are muted, Shankar.
Yeah, I have just some naive questions. So what kind of protein is this Rho A?
Hello?
Tathagata. So first of all, I mean, it was a wonderful presentation. Congratulations to you. Lot of work. And so my two very quick questions. What is the protein Rho A?
So Rho A is it's a small GTPase.
GTPase, eh? Okay, I suspected. And the second, because you at some point of time you talked about another domain of Rho A. So I thought it is a small GTPase, but then I am not able to imagine that Rho A will have more domains. And the second question is when you talked about interactions between Rho A and the other protein, here the assumption is both are present at the same time, and but we cannot conclude that they physically interact. Is it correct?
So when the group pulled down Rho A, they could when they pulled down and isolated the proteins coming down with Rho A, they found that it's DPYSL3. They used an antibody to stain that. They had initially the silver staining, and then they looked into the antibody staining that it was indeed DPYSL3, which is physically interacting with OA.
Oh, physically okay.
Yeah. So we repeated this experiment also, as you said, that it's important that the context is important where both DPYSL3 and ROA should be expressed together. So it's not strong always. It's not that it's very strong. It's only in conditions that they show very strong interaction. But also in normal cells, like when we cultured BMSCs, and in Mike Sharma's lab, Dr. Mike Sharma's lab, there was another collaborator of mine working there. Like he was a graduate student who was helping me out because I didn't know much of cell biology. So he helped me do a pull down assay where we could see that when ROA GFP, they used ROA GFP, when it was pulled down, DPYSL3 was actually coming down along with it. So there was very mild staining of DPYSL3 in the sheet and when you do the Western, but then it was coming down. So it's physically interacting with ROA. That was observed both here and in case of what was seen by the group which reported that.
Tathagata, if you go to the C4RIP slide, Shankar's other question was about the domain.
Yeah.
So Shankar, the domain is not in ROA, the domain is in DPYSL3.
Oh, okay. So I thought that he was talking about another domain in ROA. So
no. It's domain of it's the N-terminal domain of DPYSL3 itself, which was used to.
Okay, you are talking about the domain in the other protein. Okay.
Yeah, it's just a competitive inhibition, nothing more than.
So fantastic talk, Tathagata. Congratulations.
Thank you.
Any other question?
Professor
Students? Yeah. Pradeep, are you there? Okay, so if there is no other question, I should,
Chitral has a question, I think.
Chitral has a question. Chitral, where are you? I do not see your hand. Yeah, please go ahead.
Yeah. Hi, Tathagat. Really nice presentation. I am not much acquainted with the field, so I have very few basic questions. One is that in the initial slides, in one of them that you mentioned that this GDFD bead implantation has been done. So this is something which it's a technical question from my side that so everything I believe in this developmental, I mean, you're seeing these things happen in the early stages, right? So everything I presume will be in a dynamic state. So how exactly is this bead implanted, at that particular location? And, I mean, how is it prevented from getting dislocated or something?
So again, this is something that Upendra does regularly. So, but I'll tell you just to show that I know how he does that. I can see how he does that. So what he does, or what they have done, is that what we do is we soak the beads, which are very small beads. I've forgotten exactly the material which is used. But what he does is it picks up the limb and makes a small slit, and then he puts the beads in. So that makes sure that the beads don't fall off or are not flown, or it doesn't flow away with all the fluids in the egg. So a small slit is made and then it's put in. So it's difficult. There would be a lot of false positive experiments where the beads would fall off. But with, I mean, as you keep on doing, like he does like 300 experiments, and then he would have finally a good hand of putting the beads, making the right amount of slit, and then putting it in so that it stays in the location. And the second thing is, if you were asking about that how do we know that the bead is still there or not, or if the bead was at all there, it will not fall off. So actually, when you let these embryos grow, the bead gets incorporated into the embryo. So when you section them, you can actually locate the bead. You can see a very nice circular bead when you take the sections. So that, apart from knowing where the beads you have planted, you also know when you take the sections, you can actually locate the bead. Maybe if I take you to, I don't know about this because this is their data and I've just picked it up. If you look into Upendra's data, which I have here, you might just be able to look at the bead. So it might be difficult. So here actually you can see the bead somewhere.
Upendra has done bead implantation.
Right. Okay.
So Chitral, actually what it is that you do hundreds of embryo manipulation, and you are doing it blindly. Okay, you are doing it at a stage much before cartilage formation or whatever the tissue you are manipulating.
Right.
And you do it blindly, and then you have to section those many, and then you hope by chance you will get one or like not one, like 5-6 embryos with the bead in the appropriate location. Like if you look at this particular slide, if the bead was not in the start position but little bit above or below, the experiment will have no interpretation. Okay, so it is purely a matter of chance and number. So
I was
yeah, like
curious for that reason. Yeah.
it is hard work. Let's put it mildly.
Yeah. And another thing is, you were also mentioning about the levels of ROS in the last part. No, the second part of your talk. Yeah. So have you also checked the ROS levels in your with the different levels of this PRDX expression? Have you?
Yeah, so we haven't been able to check in vivo the RO levels, because the thing that we had, I mean, I did staining to see if it's actually regulated by BMP signaling. So we have a cell line where you can deplete BMP. So there we checked the RO level, because as I showed you that BMP is regulating PRDX1, and we saw that when you deplete BMP, there is actually upregulation of ROS, and that we had shown. But in vivo, we haven't shown that because we did not have the exact setup to have the ROS experiments like the helium chambers, etc. So what happens is when you do the DHE staining, it has to be protected from the environmental oxygen and etc. So you cannot use that. So we do not have that setup. So right now we haven't used that, but we will be using that to understand how Indian HIJAB has been regulated if we pursue that further.
Okay. And my final question to you is, apart from developmental perspective, like if you look at say maybe bone injuries or some form of injury where the ROS levels are varying. So do you expect to see changes in the levels of PRDX and the other
Sorry, I guess the connection fluctuated. Just repeat the central, I missed the first part of the question. So in what context?
Like if there is an injury or something way later than the developmental phenomenon. I mean, I'm talking about say a grown individual having some kind of injury, and there you see a fluctuation in the ROS levels in the regions. So do you expect changes in the levels of these gene expressions?
Look, I cannot predict, but I think there would be some effect. But the thing is that the only in vivo experiment that I know of is, which again is it doesn't answer your question directly. It's not in an adult tissue, but they have done the same paper which I had mentioned, Morita et al. They had actually injected antioxidant proteins into the mice for several weeks after being born. So that kind of is post-embryonic development, right? So after the mice was being born, they injected continuously for 2 weeks. They injected the antioxidant proteins in mice, and what they found was it's actually abrogating hypertrophic differentiation because it was lowering the ROS levels altogether. So that is the only reference that I know of where after birth there is a problem with antioxidants and hypertrophic differentiation. But after the bone is formed, you have a normal bone formation, and then most of the hypertrophic cells are anyway gone. You only predominantly have the secondary ossification centers. So that I don't have data to predict on that. But yes, after birth, after embryonic development, there is still dependence of hypertrophy on ROS, and that has been shown with injection of antioxidants.
Okay. Yeah. Thank you, Tathagata.
Rachita, you have a question. You have raised hand. Please go ahead.
Sir, yeah, I asked my question. That's all.
No, you are Chitral, Rachita.
Oh, okay.
Rachita, you have your hand up. Do you have a question? Okay. So maybe Rachita forgot. Anyway, so if there are no other question, I would now close this meeting, my presentation. I just want to share one piece of information with all of you, that apart from PhD thesis, Tathagat mastered another art, that is how to manage free money for attending international conferences. He attended 7 international conferences in all possible continents other than South America in during his PhD thesis tenure. So anyway, with that, I would close this meeting. A big hand to Tathagat for his hard work.
Thank you.
And so I would now request everyone else other than the committee members to please leave the room. Tathagat, you need to stop the streaming, and
Okay,
you
so
also need to leave.
So I'm
Okay,
stopping the live stream now. Yeah.